as they arrive. So welcome everyone. Please can you all mute your microphone, just a bit of housekeeping. Also turn off your camera unless you're speaking or presenting. I'd prefer it if you could write questions and comments throughout the session in the chat box, and we'll try and collate these together at the end. Please make sure to write your name and affiliation in the chat box unless it's really obvious who you are from the username. I'd like to welcome you from the Cambridge Conservation Initiative. Cambridge is the hub of the largest cluster of conservation organisations in the world, and this seminar is therefore brought to you by the University of Cambridge in collaboration with the 9 leading internationally focused biodiversity conservation organisations in and around Cambridge. And it's great actually to be able to bring people internationally here using Zoom. These seminars are all recorded. They're every week. You can watch last week's panel on the website, How to Prevent Further Pandemics, and you'll be able to watch this one online in a few days. Next week on the 14th of October, we have another seminar on building a biodiversity-based economy by making use of nature, and that's taught by Professor Federica Palmer. We're usually based in the David Attenborough Building, which is an inspirational campus designed to facilitate exchange between different organisations. That's where I'm based in my PhD research, and it's really fantastic for me to be back in the building today for the first time in 7 months and be virtually back together with you all and magically transport Roland to be with us here today. So we'd like to welcome you all the way from America. Roland is a research associate professor at North Carolina State University and a director of the Biodiversity and Earth Observation Lab in the North Carolina Museum of Natural Sciences. A self-proclaimed critter chaser, Roland's career has followed free-ranging animals using techniques such as GPS tags and camera traps, and he combines these high-tech methods with traditional data collection methods in the field and also in museum collections. He's also a science communicator and a regular presenter on the Science Channel and also on the National Geographic's The World's Weirdest, so I'd be interested to hear a bit more about that. And I recommend his podcast, which I was listening to today, Wild Animals, where you can follow his fellow animal trackers on their run into the Secret Service while tracking and sedating fishes, which is a large species of weasel family in the woods. Today, he's going to be talking about cyber museums, which is obviously very current, as we seek to engage remotely within science and also with the public online at the moment. So I will hand over to Roland for his talk. Thank you very much.
Great. Well, thank you very much. Thanks for the invitation. It's an honor to come speak to everyone here. Some old friends, some people I know of but haven't had a chance to meet, and it's a bummer that we can't do this in person. Obviously, there's a lot of other changes, you know, but I wanted to try to make this as unique as possible. So I did spend last night pretending like I was on an airplane. You know, went to bed early and didn't really sleep very well, got up really early. So I was completely knackered trying to, you know, mimic what it's like to travel overseas. But then, you know, fortunately I made myself a nice British breakfast this morning. So I'm back up to speed and ready to talk to you about this research. And I wanted to start, you know, I know that there's a great museum there in Cambridge, and I work at a museum, and I want to kind of start talking about the dual missions that museums really have, and that is both scientific discovery and inspiring conservation. And you guys, you know, we do this through a variety of ways there in Cambridge and also here in Raleigh, similarly. So I really like this is the museum I work at, the Museum of Natural Sciences. And I really like our mission, which is to illuminate the natural world and inspire its conservation, because it really sort of highlights these dual missions that museums have. And you know, of course, we do this through exhibits and public programs that are, you know, meant to inspire people about the natural world. And then a lot of our discovery is through collections, research with the natural science collections. And increasingly, those collections are include sort of big data and the digital data associated with the natural world. And so that's a lot of what I'm going to talk about today. And I want to, you know, catch people up if you're not at a museum, that the collections in museums, like these skulls that we're looking at here, are really at the foundation of a lot of studies about taxonomy, about evolution. There's a lot of research you can do with the physical specimens in terms of taking genetics, doing isotopes, right? Every year we're finding new things that we can do with these specimens. And so there's a lot of use for these physical specimens. But what I'm going to really be talking about mostly today is the data that comes on the tags and data that we get from other sources like this. This is the spatial data, right? You can read where these shrews were from, when they were collected, and you can use this physical voucher to verify, you know, what species they are. And this is really the foundation for a lot of our biodiversity data, you know, used to make range maps, like you see here, of sort of drawing blobs around there. These are, you know, critical to any field guide. This is a field guide to mammals in North America that I worked on that is, you know, full of range maps that are built on largely the records from museum specimens. And these are now, you know, really being standardized through the IUCN Red List. And of course, they're involving other data as well. But it really is, for some groups, it's mostly museums. And for other groups, now we're starting to use other things. And you know, after we, you know, decades ago started doing these range maps, we started seeing that there was a lot more information that we could gather from this. And we started doing spatial modeling. So I want to talk about what we can do with spatial modeling. This is sometimes known as niche modeling or species distribution modeling. And I'll give you an example with the Olinguito, which was a species that I was fortunate to help describe from South America a few years ago. This is the Olinguito. It is a olingo, but it's a little bit smaller, and it's a different species that's found up in the mountains. And when we described this, you know, it was a new species, so we had very few records to go on. There were about 13 Olinguito specimens that we could round up from museums, dating back, you know, over almost a century. And so what you do in these niche models is you take your records of where you know they are, and you combine them with environmental data. So in this case, we used a bunch of what's called bioclim data, which is things about temperature and precipitation, these types of climate things. And we're able to create this niche model that predicts, you know, based on only 13 records, we can get a guess of where else we think they live, where else the conditions might be suitable based on this. And after we initially did this, what was great was we published this Olinguito paper, and it was kind of big news. There was this cool new species, and a lot of people in Ecuador and Colombia took notice, you know, where this guy lived. And we ended up getting a lot of pictures sent to us from citizen scientists who were like, oh, I got this picture. Is it an Olinguito? And you know, we could usually tell because you can tell by the color of the fur and the length of the fur. And so we ended up actually quite quickly adding a whole bunch of new records. And where we were able to improve that. So again, we took our animal locations, and this is what we call sort of presence-only niche modeling, because we know the species are there, but we only have those records. And we're able to create a prediction map, and this is our refined prediction map. And it's showing you sort of habitat suitability. And the great thing about this is it highlights, in this case, some examples of areas where we think there are good habitat, but we don't actually have any records. So we don't know if the Olinguitos are there or not. It seems like it's the right climate, it's the right precipitation, but it's highlights for new areas to do surveys for these species. And then we also have some description of the ecology of this animal that we barely know because it's a brand new species from the niche. And so this is just showing precipitation and temperature, the range over which this species is known to occur. And so these are the cool things you can do with niche modeling. You know, unfortunately, if you look at some of this habitat, it looks like this, right? This is cattle pasture. This is not Olinguito habitat. The Olinguito needs forest, needs cloud forest. And so this is part of the problem that we have. We've got Olinguito specimens going back to 1923, but who knows what's going on there right now, right? And so this is the real challenge that we face as trying to understand you know, the needs of biodiversity, where biodiversity lives, is that, of course, the planet's rapidly changing, right? You have the Human Footprint Index, which shows just the scale of change that humans are making on the planet, where red is a bigger impact of physical infrastructure of humans on the planet. And then, of course, you have climate change changing as well, so that we need to be able to map biodiversity more rapidly and write what's going on now, not what was going on in 1923. And so some of the priorities that I see and that I'm going to talk about today for mapping biodiversity in the Anthropocene is that we need to be, mapping out where these animal populations are, monitoring population trends to see where they're doing worse, where they're doing better. Sometimes they're doing quite well, and have develop some of these spatial models to identify what's affecting their distribution, whether it's climate, habitat, species interactions, humans, etc. These niche models can help us discover what's affecting their population. Identify newly suitable habitat, especially in the face of climate change, right? If we're thinking about the habitat that might be suitable for a species, might be further north or further up the mountain or something. And we need to start to identify where those are. And this is something that these types of models are good for. But we also have to recognize that these animals are not static creatures, right? Species adapt, and sometimes they adapt quite quickly. So there might be some changes over time, sort of non-stationarity in the ecology, some local adaptation of the species. And it could also vary across populations of the same species, right? So you could have one population that adapts to humans in one way and another that doesn't. And so even sort of looking at the whole species level might not be enough when you have things changing within the species range and maybe in different ways. And so these are some of the challenges, I think, for mapping biodiversity in the Anthropocene. And this is why, to address this, we need a lot more data. And basically, museum specimens cannot keep up with the pace of change on Earth, right? So this is just showing the mammal specimen collection for American species, 1875 to 2015, and you can see it's declining. I mean, we're still collecting, but if you divide this by the 400 and some species of mammals by every year, by every state, right, we hardly have any information at all. And so I do want to emphasize that specimen collection is still super useful. It's really important. What I'm talking about today really is spatial ecology. So we still need to keep collecting those for spatial ecology and for all the other uses that specimens have. But there's a lot of other ways, especially for mammals, that we can collect useful spatial data at the scales that and the temporal resolution that we need to. And so, what this really boils down to is we need more dots on the map. This is a map of where we've run camera traps in North Carolina. And this gives us the spatial resolution to ask some of the questions I was just talking about, and the temporal resolution to evaluate on a regular basis how things are doing. And so what I'm going to be talking about today is how born digital biodiversity collections can meet this challenge. Particularly, I'll be talking about animal tracking and camera trapping. And what I mean in born digital is they don't come from a physical specimen that then has a location written down. They're digital from the beginning, right? It's a record of where the species was from a GPS collar, or it's a camera trap picture that recorded a species there. And so I want to talk about how we can use this for this sort of analytical and practical purposes, but also how these can inspire conservation. Because remember, museums are not only about making discoveries and gathering this knowledge, but it's also about getting people inspired enough to do something about it. And I think there's routes for that in this born digital data as well. And also I'll get to a little bit of what I think is really a bright future for this field is data integration, of combining these data together for a more comprehensive model. And because really these are sort of different ways of putting dots on the map. And if we can account for the different observation processes of these different data collection processes, then we can start to combine them and combine their strengths to learn more about what's going on. So that's the introduction. Now let's jump into some animal tracking. Here's me in Panama in 1996, starting my career as a critter chaser, running around on the rainforest floor with radio tracking device, chasing kinkajous. After about a year, I got about 247 fixes on this one animal. This is kind of typical. A lot of work. It gives you a general idea of where this animal lives. Fast forward to recently, I think this picture is from 2016 or 17, with the kinkajou with the GPS collar. And this is the type of data that we can get. And this is just 2 months, 30,000 fixes. The black lines are connecting the dots to show you exactly where this animal was going. In this case, this is overlaid on canopy height. So you can really see how some of these areas, canopy height's really important. They're really using the higher canopy. And you can just see with the incredible resolution, there's so much more going on here that we can learn about this species that reflects on their habitat needs, on their social organization, on their cognitive abilities, all kinds of things that you can learn from this high resolution tracking data. And this has really been the trend. If you look at over time animal tracking studies and the different technologies used, from old school VHF when Bill Cochran invented wildlife telemetry by putting a radio beacon on a rabbit and a duck and got, you know, a dozen fixes per animal, to, you know, efforts to get sort of, you know, maybe like my study in the 90s, I had a couple hundred fixes per animal, but now we're up to 1,000, 10,000, even up to 1 million fixes per individual animal. So we're really not guessing what they were doing. We know exactly what they were doing. And it's a really rich data set. We're really entering the golden age of animal tracking, where it's no longer, you know, sloughing around in the forest, for hours to get a couple locations, but it's getting much higher resolution of what these animals are actually doing. We have designed a database going back to our early days working in Panama, where we started getting more and more data, and we needed a way to handle it. Excel didn't work for this anymore. And we also wanted to be able to share it, right? So that others can use our data. And so we created the MoveBank database, Martin Mikulski and I, which has now grown incredibly. And we sort of started, well, let's just use it for ourselves. And then we had a bunch of students, and we were able to you know, manage with support from a number of agencies, and especially the Max Planck Institute, to make this a community resource. And so these dots are showing everywhere that somebody in the world is using MoveBank. All sorts of scientists use it, not just myself. And the great thing is that they have complete control over their data. Whether they want to share it or not is up to them. In this case, the gray dots are studies that I don't have access to, but I could ask for it. And so MoveBank has really grown. I think we're up to about 2 million locations now of individual animals that are in there. And again, I say we, but it's not my data. I cannot access this data unless you give me access to the data. So it's steadily increased. We have many dozens of live data feeds coming in from satellites and from cell phone networks. And again, the data owners really control access. So you don't have to share data, but if you want to, we make it easier. And we have standardized format. You can publish it, get a citation, and then others can cite you when they use it. There's now starting to be a number of groups that are using MoveBank to bring together data. This was a science paper recently that led by Marley Tucker that brought together a bunch of animal tracking data to find that animals move a lot less in areas where humans are, which was pretty interesting. And I want to give you sort of a view of what some of this animal tracking data looks like, and just how awesome it is, and how many different species we have. And so this is an animation of a stork in Europe, a bunch of storks. These are white storks, and they're migrating south. This is real tracking data from GPS trackers. And you can see these storks right in the fall. They're making this precarious flight over the Sahara Desert, which must be terrifying, and then down into the fertile grounds of the Sahel. And of course, everywhere they land, there's other species, right? So here's some wildebeest in East Africa that are doing their thing, and all sorts of interesting movement on the ground. And some of these storks, amazingly, keep going all the way down to South Africa. Of course, they're running into other interesting species like African buffalo and elephants. And so these are, again, real data that are accumulating in MoveBank that are showing, you know, just how dynamic the planet is, how much animals are moving, and how much animals really connect the planet by moving, you know, globally and locally as these storks, you know, return north back to the breeding ground for another year in Europe. So this is the kind of thing that gets me really excited. The amazing stories that are in here of these animals, the amazing amount of information and inference that we can get from this kind of thing. And, you know, with this animal tracking data, we can build resource selection functions that are very similar to species distribution models. So the types of things that I was talking about earlier with the Olinguito that was kind of broad scale across their range with museum specimen records, we can use, we can do in tracking data at all sorts of higher resolution scales. For example, let's say you have the home range that might be occupied over weeks or months, and you have these points in red that the animal actually used. And then you can generate a bunch of random points, places that were available that they didn't use. And you can make comparisons and say, well, what was special about the places that they used? What was a habitat that they're selecting for? And you can zoom in even more and do this at a daily scale and say, here's a trajectory that an animal used. Why did it use that? Let's make a bunch of other paths that follow the same distance and the same turns and see what's different. Again, sort of gives this really concrete way of making these comparisons. And then finally, these step selection functions. You can take each movement, each step. Well, let's say you get a fix every 15 minutes. Why did he go there? Or, why did the animal go there when there are all these other available points to it that it could have gone to based on what we know about how far this animal can move in this amount of time? And so these are really powerful functions. We're doing some work with this now. I'll give you a little preview of some data we're working on from Panama, where we tracked 4 different species. So 2 species of monkeys, kinkajous and coatis, which are procyonids. They're in the raccoon family. But the interesting thing is all these animals eat fruit. And at this 2-month period, when we focus in, they're mostly all eating the same species of fruit, which is a species of, it's called dipteryx. And so we were able to map those dipteryx using drones. And those are the little pink dots you see. And now you can see the animal tracking data of the animals moving around. Each species is a different color. And you can see the tracks, you can see how they're moving around. We can use the fine resolution data to see, right, when they visit a fruiting tree. We can see these cluster points to infer that they're probably using that fruiting tree. And we can look at all sorts of details about how the different animals have different routes, and looking at how they're using the habitat differently, and even trying to infer some stuff about what they know about the environment and how their cognition might be different. Yeah, so we're still working on this. We haven't published any of this data yet, but we've got some pretty interesting data looking at the habitat selection of these frugivores with a bunch of canopy data from LiDAR. And we can see, interestingly, that the coatis and capuchins are quite similar. And the spider monkeys and the kinkajous are different. So we're seeing some interesting, some ways that they're quite consistent. Interesting, they all were really concerned about sort of paying attention to the total elevation change. This area in Panama is quite hilly, and interesting, even if you're in a tree, you still got to climb the hill, right? And so they all really selected to minimize elevation gain in their movements, which is the same thing I would do if I was walking around there. So that was interesting that was sort of uniform across species, but then there's some other interesting differences that we think relate to some of the finer scale differences in the way these animals move. So I'll give you one more example of some tracking data from high resolution tracking. And these are fishers around Albany, New York. This is with my grad student, Scott LaPointe. And fishers are medium-sized predators. They're bigger than martens. They're famous for being able to kill porcupines. And they eat rabbits and squirrels and all kinds of mice. When I was in graduate school, I was really like fishers, and they were this like icon of the wilderness. They were sort of the old growth species, the animal that needed this mature forest. And so I was really surprised to find them in suburban Albany when I was working at the New York State Museum. In these forest fragments that you see right there, right, if you look in the background, you can see the mall, the university, and the downtown, and these neighborhoods. There were fishers in there. And we were shocked. They had moved in. And this is part of what I think is like the adaptation, right? Fishers 50 years ago were not in this habitat. They've adapted, and they're now using suburban habitats. And the tracking data really gave us some unique insights into how they were doing that. And we used really high resolution data. So what I'm showing you here is if we had tracked every 6 hours, or if we had tracked every 1 hour, this is what it would look like. All right, so we've subsampled the data, and I've highlighted 3 points. There's that lake up there, number 1. Looks like they use that lake sometimes. There's this area number 2 that maybe they used at 6 hours, but at 1 hour it doesn't look very important. And then there's this area down there at 3 that they visited briefly that didn't show up in the 6-hour data at all. And you can see, just for context, sorry, the dark squiggly line is a river, and the lighter lines are roads. So it's a very developed area. Well, we actually had locations every 2 minutes, and you can see they never visited that lake, right? And number 2 was a super important corridor for crossing a very busy road. And then number 3, they visited a couple times. And we went there to 3. We're like, what's so special about this place? And it was this crappy culvert. It was a drainage culvert that was half filled with dirt. But you can see the fishers going back and forth using this because it was a safe way to cross under the road and not get hit by a car. So a really cool behavioral adaptation of these fishers. And you can see they're using these corridors to connect forest fragments. We also found that they were using some of these amazing sort of unclaimed habitats at night, right? So they're using the cloverleaf of forest in this highway off ramp, and using golf courses at night when no one's there, but there's probably lots of rabbits and squirrels. Now, it was really fun tracking these fishers and learning about these individual animals. And at the time, there was a blog at the New York Times called Scientists at Work, and I sort of talked about some of our work in that. And this got me thinking about these details that we learn as animal trackers. We get to know these animals individually, and their lives are pretty amazing. And we had this sort of privileged look into it. And this high resolution data is giving us, a much more detailed look than we can ever get before. And so I've started thinking about, can we turn these stories into narrative that get people excited? Like, let's share these stories. So it's not just the animal trackers that get them and, sort of share them around the campfire, but share them more widely. So I started the Wild Animals podcast, where I interview another animal tracker and have them tell the story of a particular animal. So they're crazy stories about animals told by the people who study them. These are some of the characters in season 1, which is out now. And you can read about Bravo Luis, the monkey who became the leader on a rainy day, or Hansi, the stork who brought a baby to a farm. And so each one is kind of about an individual animal. Season 2 is out now as well. You can read about Pac-Man, the jaguar with 2 girlfriends in 2 different countries, or Jon Snow, the marmot who died and came back to life again. And I'm looking for season 3. So if you're an animal tracker and you like telling good stories, or you know somebody who knows animals this way from tracking, again, wild animals, there's other podcasts for stories about your dogs and cats. But if you've got a story you want to share, or know somebody I should hit up, let me know, because I'd like to get season 3 going. Okay, so I'm going to transition now a little bit more to camera trapping and put this in perspective of animal tracking. That really, these are 2 ways of studying animal movement, right? You've got the tracking shows you the trajectory an animal moved through, and the camera, shows you some places where an animal occurred. Usually, you don't know the individuals, so it's kind of 2 sides of the same coin. But the nice thing about cameras is you get often a whole community of animals, and you can come up with a priori study designs to really ask research questions, right? So here is a study we did. We wanted to know, what's the effect of hunting and hiking on wildlife? And so we looked at 32 parks across 6 states. Half of them were hunted and half of them were not hunted. And we tried to control, so they were very similar. They were sort of paired. So we'd find like a state park that was not hunted and a state game land that was nearby that was hunted. So we have that comparison. And then we have cameras on trail, off trail, on trail, near trail, and off trail to look at the effect of hiking on wildlife. And the nice thing about cameras also is we have explicit measure of search effort. So we know how long a camera was out for, unlike some of the museum data where you don't really know or what the effort was. And so we can say absence. We can say, oh, we put a camera there for 3 weeks and we didn't detect a species. So we can say presence, absence, which you see on the left. Or we can use measures of relative abundance. We can say, well, at this site, we got 2 pictures in 2 weeks. And at this site, we got 25 pictures at 2 weeks. So we know this site is more important, is better habitat for a species. And so we can use different models. And we have this for the whole community. We're able to work on all these different species of mammals that we get. And what we found, this is a little video that shows camera trap data from Shenandoah National Park, Virginia. You can see a lot of hikers, but bears, right out there as well. There are a lot of bears. You want to see bears, Shenandoah is a good place to go. And what we found is that habitat, so aspects of forest cover and some of the bigger scale habitat measures were more important than recreation in the models for most species. Hunting was correlated with fewer deer, which is the species that's mostly hunted. So hunting did have an effect, although there were still plenty of deer everywhere. There was actually more coyotes at these sites. And some species avoid the busy trails. So bears and bobcats, if the trail had a lot of hikers, they avoided it. Red foxes actually preferred the busy trails. So that's some of what we were able to derive from this work. Now, camera traps really scaling up. The papers are going up, increasing on a regular basis, right? It's a super useful tool, and we're using them at larger and larger scales. We're starting to run into the problem of, well, how do I deal with all these pictures? These digital cameras get so many pictures that it can quickly overwhelm you, especially if you're running hundreds of cameras. And so working with Bill McShay at the Smithsonian, we created the eMammal database at the Smithsonian to manage these data and review them and still have good quality data to allow us to work with both field technicians and citizen scientists. And we're working now on a new project called Wildlife Insights, which is a partnership of a bunch of different institutions you see here, including ZSL. Basically, we're trying to work together to create a database that is more useful and that has artificial intelligence to help automatically identify the animals. You can see that going in this little video clip. Google is helping us with that. And so in the next year, this will release to the public, and we'll start moving our work from eMammal over to Wildlife Insights. So with this infrastructure, we can now move to citizen science camera trapping, which is great because it allows us to increase our sample effort. And so this is a project that we've done in North Carolina with support of the wildlife resources. We were able to cover all 100 counties in the state. So this is a map of North Carolina. And you can see the scale over which we were able to study. We did get cameras in all all counties. And this was, again, working with the volunteers. They were able to check out cameras from libraries. So we would loan the cameras. We would distribute the cameras to libraries. Once they'd gone through our training, they could go to the library and check it out, run the camera, and then send us the pictures, basically send us the data after they had gone through it. So we were able to sample over 4,000 locations in 3 years, which, just to put this in perspective, I went to Google Scholar and just looked at the last 10 camera trap papers that were published, and they averaged 99 sites, ranged from 25 to 221 sites. So that's what's being published right now. We've got really, really rich data set. We had 106,000 animal detections of 39 mammal species. To put this in perspective, if you look at other available data, iNaturalist, which we'll talk about here in a little bit, where citizens can take pictures with their phones and share, they have 6,000 records of mammals in North Carolina. All museums across the world at all time have 15,000 records. And for bobcats, there's 27 on iNat, 27 on museums. So again, we got almost 500 bobcat detections, so way more than has ever been found. So a much richer data set just in 3 years. And 106,000 mammal detections compared to 15,000 of all time in all museums. So a really rich data set. I'll give you a little bit of examples of how we're using these to answer questions. One question was, well, how are they adapting to humans? How do humans affect wildlife? And so we set cameras with citizen scientists in urban and wild areas. So we had some volunteers, setting them right in their backyard, and some set out in the small forest fragments in rural and suburban areas, and then some were really out in the wild parts of North Carolina. And we found the urban areas were loaded with wildlife. Some species were more common in wild areas, but the overall levels of diversity and abundance were similar, if not higher, in the suburban landscapes. And it was this green space that was most important. So if there was green space, we had more wildlife there, which shows that a lot of these species of mammals, it's kind of an urban paradox, right? That you would think that in the urban areas, there'd be fewer mammals, but there's actually more in some places, in some cases. So that was a really robust study. That was actually, this one was across two different cities. We found the same pattern. There's some other work that's coming out just recently that showed some similar cases. So it's a bit of a paradox. We could talk more about that maybe in the questions. We're also now working on statewide maps of relative abundance. You can see here, here they are for 4 species. Bobcats really avoiding the developed areas in the middle of the state. Coyotes more common out in the mountains. And one of the interesting things that we've started looking at with this data is, can you have one species model across a big area like this? Or is there local adaptation? Do deer respond differently in the coast than in the mountains? And to do this kind of thing, you need a lot of data, which we have now. And we've been working with these spatially varying coefficient models, which look for these types of differences. And we found for raccoon, bobcat, and coyote, it didn't really matter. A global statewide model was more or less good. Sometimes, I think the coyote and raccoon, breaking it into thirds was a little bit better. But for deer, we found quite a lot of non-stationarity in that they respond differently to forest cover in different parts of the state. So this is showing you that in the red area, they respond positively to forest cover. If you have more forest cover there, you're likely to have more deer. And out in the white and blue areas, it's actually a negative response. And so this shows you that if you would have had one statewide model, these probably would have balanced each other out and you would have found no relationship. But now we're finding that there actually is a relationship. We're starting to explore what might drive this, whether it's subspecies differences in the deer, or maybe something different about the forest in these areas. And so this is pretty cool that you can use these models now to find some more nuance about the response of these species. And this is work by Brent Pease, a grad student at NC State. I'm gonna skip this. I'm running a little a little slow. But so I want to talk about the potential of citizen science, not only to collect more data, but also to engage with the public, right? That not only do we want them to send us our data, their data that we can use, but we want to give them results and feedback. So we've been doing this with our Canada Creators North Carolina project, running social media accounts, right? Sharing animal pictures. It's so easy. They're great. This is the great thing about camera trapping is you get lots of great pictures. And so it's fun to share these animal pictures. We've also done a bunch of videos and live webinars to try to connect with them, share the results of the project as we go. A few of these have gotten quite a lot of hits. Some of them get fewer, but they're still, I think, useful engagement tools. We've also been creating some interactive data products, so trying to make the data more accessible so they can change the species, see where they live, see what time of day they're active. We've also been working with Stephanie Shuttler to bring these into school curricula. So we've developed, you can go to eMammal Academy and see curricula that we've developed so the kids can run cameras. We've got a lot of great data from schoolyards, from like the woods, little bits of woods near schools. And there's a lot of wildlife there, and the kids really enjoy looking through these pictures. And we found that they can really get us useful data, not only in the United States, but Stephanie was able to work in some of these other countries with school kids. And they got some amazing pictures, not just in the national park, because they didn't run the cameras there, but in the areas near their house. We got jaguarundi in Mexico. We got puma, and the super endangered Grevy's zebra, black rhino. India, we got gaur, dhole, and even some tigers. On this road that during the daytime you get pictures of the village people walking back and forth. And they were even, really able to connect with, make a local impact. The Mexican kids working with the museum there in Mexico had a showcase with the local politicians to share what they found, which is really cool. The Indian school kids have gotten lots of media attention, so that's been really great. We're also trying to use these stories to tell pictures, kind of like we do in the blog post and the podcast with the animal tracking. And so I've got a book that came out in 2016 that tries to tell science stories using some of these great pictures from all around the world. We've got a Twitter feed where we share some camera trap pictures from a fox den that's pretty popular. And we developed this a little bit more in this YouTube video where Stephanie and I sort of tell the story using the the camera trap pictures, but also having us kind of, narrating the story. And this video in particular has really taken off. It's got over 1 million views. It's the most watched video on the museum's YouTube channel. So I think this is, if you find situations like this when you camera trap, it's a great opportunity to tell a story. And we're finding that we're creating advocates for nature. So we've been doing some before-after surveys of our camera trappers, and we find that they're increasing their connections to nature. They're actually increasing their science efficacy, and they're becoming advocates for conservation. They're more likely to teach others about local mammals and conservation. So Bill McShay at eMammal and the Smithsonian, liked our statewide project, but he says we need to scale up. We need to sample the whole country. And so we've been working on the Snapshot USA project with scientists, having scientists run cameras across all 50 states. We had great success last year with 127 participants in all 50 states. The data is all together. It's now in a publication in review with all the scientists as co-authors. So this is the payoff. You run your own cameras, send us the data. Let's collaborate the data, get it all together and clean, and we'll publish it every year as an annual national census of camera trapping. We're analyzing the data right now. There's a lot of cool discoveries to be made. We also have Snapshot 2020 underway right now. Some folks are sharing pictures on Twitter under Snapshot USA, and we're interested in helping others use this model locally. We're talking about possibly Snapshot Europe in the future because I think it's got a lot of potential. And so I hope you see that animal tracking data and camera trapping data are both really expanding. The coverage globally is pretty good for animal tracking. On the camera trapping side, we still have a way to go, but we're just now starting to bring people's data sets together. I want to briefly mention iNaturalist, because here you can see the increase of camera traps and museum specimens. iNaturalist is growing like crazy, like 2,000 to 4,000 records every week for North American mammals. And how accurate is it? Well, we can check, right? Here's a picture that someone took, and you can see it's been identified by 4 or 5 people as an eastern gray squirrel. And we're working with mammalogists to go through these records. There's over 5,000 records before 2020. Sorry, 500,000, yeah, which is nearly the size of the Smithsonian mammal collection. And what we're finding is that most species are accurate. Some need careful review, like the weasels in particular, and some are impossible, right? There's some you just can't tell apart. But I think if we know this, there is a bonanza of records coming in through iNaturalist that we should really be paying attention to. So I think, what's coming in the future, and a lot of folks are working on this, that I think is really exciting, is this data integration to combine the animal tracking and the camera trapping and the museum specimens and citizen observations to get a richer view of what's going on. This is an example in North Carolina we're working on, comparing camera traps and hunter observations. And so I hope you'll see, just to kind of wrap up, that for biodiversity conservation, we need to know what to do, right? And that's reaching the mind, and that's something museums are very good at as researchers. But we also have to have the will to do it, and that's what I'm kind of getting at, like touch the heart. Like we need to help people connect with nature, and especially in this pandemic, we're connecting more and more digitally, and so we need to find ways to do that. And it's our job as zoologists to really find these amazing animal stories that will change the world. And sometimes those are told in peer-reviewed graphs and publications, but sometimes they need other platforms. And we should be looking for these and trying to share these stories, because we know these animals better than anyone, and others want to know it. So we need to follow really Attenborough's lead to get these stories out there. And so this is my sort of view and vision of let me get this one going. How camera traps and animal tracking can help illuminate the world, the animal world, and hopefully connect both minds and hearts. So that's the end, and I'll take any questions. Thank you.
Thank you very much. That was really, really encouraging. I particularly enjoyed the tracking visualizations you showed. It was great to see that while we're all stuck at home, animals are still on the move and they're actually interacting with each other. Really,
they are. I should put a plug. I forgot to mention, we have a app called the Animal Tracker app, that you can see live animal tracking data from all sorts of different species all around the world.
That would be fantastic to look up later. And also, having been involved in citizen science before, the iNaturalist number of records that you had compared to museums, that was incredibly interesting. I've actually kind of changed my mind and said that I'd be happy for people to ask questions live because we're not a huge group. So I'm just scanning to see if anyone's raised their hand on the participants, which it doesn't look like they have. So, oh, here's one from Lauren. Lauren, shall we unmute you so you can ask your question live if that's going to be too frightening? Oh, all right.
Hi. Hi, can you hear me okay?
Yeah, good.
Yes, we can hear you.
Hi, so I just posted something in the chat. I'm the curator of the University of Cambridge's Herbarium. I was just going to kind of shout out to everybody that the university not only has this extraordinary zoological museum collection, but we have the botanical equivalent as well. And in many places, particularly in the US, herbaria and zoology museums are combined into natural history museums. So many of the things that Roland was talking about are analogous to what we do in botany as well. So please do get in contact if people are interested in collaborating. We woefully underuse our collection, and the US, as Roland showed, use their collections, their herbaria and their museum collections so much in teaching and research. There's some really inspiring stuff going on, and Roland gave a brilliant overview of a huge amount of that work, but there's so much and so much resource out there. So yeah, sorry, I just did that horrible thing of more of a comment than a question, but just a reminder to everybody, there's loads more here than what you realize.
No, that's great. So how do you get good, I guess, what's the plant stories? Like how do plants tell stories compared to animals? I mean, we can see the animals moving around and personalize them. And there's some charismatic plants out there, but
there's loads more than people realize, for sure. This is a whole subject for another talk. People talk about plant blindness. There's some work at the moment about plant awareness disparity being more of a scientific term to be an appropriate term to be using for exactly that. But when we start engaging with students, undergraduate students, school students, members of the public, don't have problems engaging them with plant stories at all. But I promise you, there's so many human connections, ethnography, cultural traditions, plus charismatic taxa. We can work on that, but maybe we need to convince some of the zoologists a bit more that plants are cool too. Thanks, Mike, for your shout.
Plants are cool too. I definitely agree with that. Are there any other questions from people? Yeah. It's not just while people are thinking. I had one question about all of the wildlife kind of spotting, so dots in the graph that you're doing. You're kind of bringing all those things now indoors rather than necessarily having your pair of muddy boots, and that's often very much the experience of being a conservationist and being an ecologist, is going out and actually physically having that touch and interaction yourself, which often allows you to gain much more of the context that you necessarily would than some of these citizen scientists who may be indoors analysing some of this information. I was wondering if you could reflect on that a bit and how we maintain that connection to the physical world, especially in the context of having AI to identify different species, where many people would have cut their teeth doing identification in the field kind of in real time. I was wondering if you could talk a bit more about that.
Sure. Well, I think, we still need to get outside to get pretty much all this, right? I mean, you can put a tag on animal and then let it go, and then you can sit at your desk and just watch the data come in. But actually, from that perspective, I have learned what's super useful, and one of my favorite things to do is to get the data from an animal, like where they were yesterday, and walk in their path. And you actually can learn so much by just walking through, where an animal was. With the fishers, it was great because we could see their tracks in the snow sometimes. And so we could literally know exactly where they were. So that's one useful thing. And then, all these iNaturalist data is from people going out and enjoying nature. And, that's great. Those are citizens going out. And it's really encouraging them to go out. And I think the AI is actually a huge benefit in this regard, because yeah, someone mentioned the Seek app. Like, I can go out and identify mammals without an app, but I'm terrible on mushrooms, right? But now I'm starting to learn them because, you know, it's fun to use the Seek app, and they tell me what it is, and now I'm starting to, you know, learn some more about those. So I think the AI really lowers the bar for people who aren't already experts. And then on the camera trapping side, you know, you're still setting cameras. Now, when we had the volunteers set cameras, it's true that I didn't get to know a lot of those places. And in this, you know, Snapshot USA project, I don't get to visit all these places, unfortunately. I'd like to do a road trip and check them out. But, you know, I'm setting my own cameras and I'm getting to know those sites. And there's also something very powerful. It's often referred to like Christmas morning, when you get your memory card from your camera trap and you plug it in and you get to see, you know, a place that you just were, what was going on there in the last month, what animals were walking by that, and what were they doing. And it really gives you a deeper appreciation for that place and a much better understanding of sort of what's going on in that forest or whatever, by looking at these pictures.
And you had a question that you wanted to ask.
Hi, Roland. I had a quick question for you. I really enjoyed your talk. Want to get your perspective on, you know, you're focusing on the movements of these animals, but we have the technology now to collect all kinds of environmental data, temperature, humidity, even record the communication signals that these animals are producing, and just like what that's going to open up for, you know, in the future. I know what the state of the art is. I know a little bit about, you know, tracking big migratory fishes and where they're getting really interesting data sets, but I just imagine how quickly this technology is moving, how much better your niche models could become if you had environmental data collected directly from the individual organisms themselves.
Yeah, no, that's a good question. There's a lot of interest in that, especially in the marine world, because to have these animals with a, you know, a temperature and maybe a saline sensor, and they're diving, they're going deep, they're going all over the place, and so that's really important. And then, you know, we can get, you know, there's been a huge boon in remote sensing data that can tell you, you know, the temperature and the cloud cover and maybe the habitat type, right? But it's usually pretty big scale, right? It's you know, usually many kilometers or something like that, whereas the animal's telling you what they're actually experiencing right there. And so I think it can also be really useful from that perspective. You know, the other one that people have been playing with that's still pretty limited, but I think has a lot of potential is you know, a camera on the animal, or acoustics on the animal, like you mentioned. There's still a lot of challenges because, you know, if you run a camera, if you have 25 animals, you know, collared, and they all have cameras running 24 hours a day, oh my God, you're going to get so much footage. What are you going to do with all that? And so that's really a challenge. And then it's really, you know, video is really heavy. So how do you get the files back? How do you process it? So there's definitely been some nice studies on it, using those tools. I think the acoustics right now is really starting to take off. And you know, so many interesting questions about animal communication that you can do with a microphone actually on the animals. There's going to be a lot of exciting stuff out of that.
Yeah, think
There's
about
some really cool work with bats, looking at feeding, you know, from their calls. So lots of cool stuff.
Yeah, I mean, I'm aware of like the Audio Moth, the open source platform, you know, how cheap those are now, and these autonomous recorders. So that technology really seems to be taking, and I agree. I think the audio would be a much better, for a lot of logistical reasons, data set to add. But I just think of the communication, you know, you have a context for it, you know exactly where the animal was. I mean, you can set up autonomous arrays and record the animals, but and try to triangulate where they might be when they're producing a particular call or signal. But actually knowing where it is in three-dimensional space with the data you're already collecting, I think would really up the level of precision that would be available.
No, I'm not hearing you, Lucy.
Sorry, can you hear me now?
Yeah.
I think we have time for one more question. If anyone wants to raise their hand. Oh, there's one from Mike. Yes, Mike, sorry.
I have a question for Roland. This is a really interesting talk. I wonder whether there's a potential for a switch away from collections in museums to masses and masses of digital data, which is interesting. But but it will increase the pressure, the financial pressure on institutions and museums, because people and grant-giving bodies and the government will say, oh, well, all this stuff's old-fashioned now. We need just pictures and tracking and all that sort of stuff. Do you see that as a problem?
Well, I tried to emphasize that I don't see it as a switch away from collections. Traditional collections are still super important. We need to be collecting, the animals today to document what's going on in 2020, right? There's all sorts of information that you can get out of specimens that you can't get out of a digital record. And so I think that's really important. But you're right that we're now asking museums to do more, like not only, curate the physical specimens, but curate the digital collection as well. And that's expensive, right? That's database managers, that's computer scientists that need to be paid, competitively to get a good one at the museum versus, working for some commercial company. And so I think that's a big challenge. But the question is, somebody needs to do it, right? The field of ecology is moving in this big data world. And I think museums are a natural home for that kind of place. And, we have to make it's not a zero-sum game. It's not like you need to stop supporting collections in order to do this. I think this is a real opportunity to meet the mission of the museum with digital resources. And I don't think we should be moving away from it. We should be sort of adding that to our mission.
Thanks.
I missed a question from Mike. I don't know if you want to jump in, Mike, with your question, which then
Sure.
might be the last.
Let me just, yeah. So that's been fascinating. I was asking about museums, Roland, and I think the last Ed's comments were fascinating, because what I think you can do with your materials is you can add life to existing dioramas and existing exhibits. So a static classic East African diorama exhibit in a museum suddenly now becomes supplemented by live migratory imagery from the Serengeti. And that could be really powerful. And museums are always looking for the next thing to bring the punter back, to bring the family back. And I think you could provide some really scientifically fascinating and personal, quite enthralling data to that. So congratulations.
Well, thanks. Yeah, we've worked with some museums. MoveBank is kind of becoming the place to go to find animal tracking data. And so we've been, but again, our role is to connect, right? And so if you come and you say, we're looking for data from the Serengeti, we can say, here's the people who have data from the Serengeti, and they can share it with you on MoveBank. Then you get a standardized format, so it's all in the same consistent, metadata, and it's easy to deal with. And so we've worked with a couple different museums already. We've made some of the animations that I showed you today ourselves.
They're neat, yeah.
But again, it's also about like finding we have a big project with National Geographic right now where we've sort of worked with getting permission from the different users and getting them big chunks of data to use for some animations.
No, it's fascinating. I'll follow up individually on how you could identify the Dipteryx trees by drone. That's quite neat.
Because they have purple flowers during the flowering season.
So that's easy then. They're flagging it up. You see, you're not plant blind.
Oh, totally. Oh, yeah, no.
You've got 50-50. Brilliant. Oh, fascinating. Thank you.
Great. Thanks.
Thank you so much, everyone. I think we need to bring it to a close, but that's been a really fantastic talk, really, really interesting opportunities for us all to still get outside while also collecting that really important digital data. I've really enjoyed listening to you today and actually having you beamed in from the States. It's been a real pleasure. I should just also highlight that one of our participants, Vivek, has highlighted that there's another conservation talk that's going to be happening on the 9th of October at 4 o'clock. He's pasted a Zoom link in the chat, so do save that before you sign off. And yes, thank you everyone for joining, and I hope to see you next week for the next CCI seminar. Thank you very much.
Great. Thanks everyone for coming.